Chapter 6. Why Some People Have Blue Eyes. If you create a global map of eye color, but put aside the migrations of peoples in the last few hundred years, you will see that light eyes, blue and green, are common only in a region centered on the Baltic Sea in northern Europe. Meanwhile, almost everyone else in the world has brown eyes, and there's good reason to believe that brown eyes were universal, or nearly so, prior to the emergence of this pattern of eye color. Here's the puzzle. Why are light eyes distributed in this peculiar way? To understand this, we need first to consider how culture has shaped genes for skin color over the last 10 millennia. Much evidence now indicates that the shades of skin color found among different populations, from dark to light, across the globe represent a genetic adaptation to the intensity and frequency of exposure to ultraviolet light, including both UVA and UVB. Near the equator, where the sun is intense year-round, natural selection favors darker skin, as seen in populations near the equator in Africa, New Guinea, and Australia. This is because both UVA and UVB light can dismantle the folate present in our skin if not impeded or blocked by melanin. Folate is crucial during pregnancy, and inadequate levels can result in severe birth defects like spina bifida. This is why pregnant women are told by their physicians to take folic acid. In men, folate is important in sperm production. Preventing the loss of this reproductively valuable folate means adding protective melanin to our epidermis, which has the side effect of darkening our skin. The threat from intense UV light to our folate diminishes for populations farther from the equator. However, a new problem pops up because darker-skinned people face a potential vitamin D deficiency. Our bodies use UVB light to synthesize vitamin D. At higher latitudes, the protective melanin in dark skin can block too much of the UVB light and thereby inhibit the synthesis of vitamin D. This vitamin is important for the proper functioning of the brain, heart, pancreas, and immune system. If a person's diet lacks other significant sources of this vitamin, then having dark skin and living at high latitudes increases one's chances of experiencing a whole range of health problems, including, most notably, rickets. A terrible condition, especially in children. Rickets causes muscle weakness, bone and skeletal deformities, bone fractures, and muscle spasms. Thus, living at high latitudes will often favor genes for lighter skin. Not surprising for a cultural species, many high-latitude populations of hunter-gatherers, above 50 to 55 degrees latitude, such as the Inuit, culturally evolved adaptive diets based on fish and marine animals, so the selection pressures on genes to reduce the melanin in their skin were not as potent as they would have been in populations lacking such resources. If these resources were to disappear from the diet of such northern populations, selection for light skin would intensify dramatically. Among regions of the globe above 50 to 55 degrees latitude, for example, much of Canada, the area around the Baltic Sea was almost unique in its ability to support early agriculture. Starting around 6,000 years ago, a cultural package of cereal crops and agricultural know-how gradually spread from the south and was adapted to the Baltic ecology. Eventually, people became primarily dependent on farmed foods and lacked access to the fish and other vitamin D-rich food sources that local hunter-gatherer populations had long enjoyed. As a consequence of this combination of living at high latitude and a lack of vitamin D, natural selection kicked in to favor genes for really light skin, so as to maximize whatever vitamin D could be synthesized using UVB light. Natural selection would have operated on many different genes to favor very light skin among cereal-eating Baltic peoples, because there are many genetic routes to reduce melanin in our skin. One of those genes is called HERC2, which is located on chromosome 15. HERC2 inhibits or suppresses the production of a protein by a nearby gene called OCA2. Suppression of the synthesis of this protein, which occurs through a long and complicated set of biochemical pathways, results in less melanin in people's skin. However, unlike other genes that influence skin color at other places in those pathways, HERC2 usually causes light eyes because it also reduces the melanin in irises. Blue and green eyes, then, are a side effect of natural selection favoring genes for lighter skin among cereal-dependent populations living at high latitudes. If cultural evolution hadn't produced agriculture, and specifically techniques and technologies suitable for higher latitudes, then there would be no blue or green eyes. In all likelihood, then, this genetic variant only started spreading within the last 6 millennia after agriculture arrived in the Baltic region. The point of this example is this. Cultural evolution can shape our environments, and consequently, it can drive genetic evolution. In cases of recent culture-gene coevolution, in which the relevant genes have not spread to replace all or most competing genetic variants, we can isolate the causes and effects, and sometimes even finger the specific genes being favored. This is important because some researchers have argued that culture could never be strong enough for long enough to drive genetic evolution. Recently, however, new mathematical models and mounting evidence from the human genome provide a clear, if only preliminary, answer. Not only has culture driven specific genes to high frequency in some populations in the last 10 millennia, but in fact, sometimes cultural evolution can create selection pressures more powerful than seen elsewhere in nature. Sometimes culture catalyzes and drives more rapid genetic evolution. To be clear, this book is about how culture drove genetic evolution during the emergence of our species. It's about human nature, not about the genetic differences among current populations in our species now. However, I'm going to use the fact that culture-gene coevolution continues today, with many culture-gene interactions still in progress in our species, to illustrate the power of culture to shape the genome. Aside from this chapter, I'll only occasionally be able to link specific genes to the culture-gene coevolutionary processes described. This is for several reasons. First, many of the coevolutionary processes I'm focused on are completed, such that the traits under selection don't vary across our species. This means that we can exploit the variation among populations, or what is known about the movements of populations around the world, to infer the underlying causes of the spread of particular genetic variants. Second, many human traits are influenced by many genes at different locations in our chromosomes. This makes it quite difficult to finger specific genetic variants, since any one variant contributes only tiny effects. Finally, this enterprise is really just beginning, so while the broad outlines can be discerned, much more work needs to be done. Let's consider another example. Rice wine and ADH1B. In mammals, the alcohol from rotting fruit and other sources is broken down by enzymes produced by alcohol dehydrogenase, ADH genes, and eventually processed into energy and metabolites in the liver. However, if the rate of inflow of alcohol, ethanol, into the liver is too high, then it overflows, going into the heart and then spreading throughout the body. Intoxication ensues. Most primates aren't particularly good at processing alcohol. However, about 10 million years ago, when our common ancestor with gorillas came down from the trees to spend more time on the ground, rotting fruit probably became a more important food source, so our ape lineage evolved a higher tolerance for consuming alcohol. This ancient adaptation appears to have set the stage for much more recent culture-gene coevolution, as humans have experienced a great deal of evolutionary action on various alcohol processing genes since the origins of agriculture. Let's consider just one of those genetic changes. Between 7,000 to 10,000 years ago, the DNA on one of these ADH genes, ADH1B, on chromosome 4, flipped a bit, causing it to code for the amino acid histidine instead of arginine. The evidence seems to suggest that this new version of the ADH1B gene metabolizes alcohol much more efficiently in the liver. Perhaps more important, the rapid breakdown of alcohol produces high levels of acetaldehyde, which causes dizziness, increased heart rate, nausea, weakness, overheating, and a flushing of the skin. The unpleasantness of this flushing reaction reduces people's susceptibility to alcoholism and parallels the effects created by drugs used to treat alcoholism. Estimates vary, but possessing the booze-inhibiting variant ADH1B reduces the likelihood of alcohol dependence by a factor of between 2 to 9 times, and both heavy and excessive drinking by a factor of about 5. The more efficient alcohol breakdown performed by this variant also likely protects the body against drinking binges, but may make hangovers worse. Have you ever noticed anyone flushing after drinking a relatively small amount of alcohol? Who was it? Data on ADH1B has been gathered from around the world. It turns out that the booze-inhibiting variant of this gene is distributed rather non-randomly. One illustration shows that the hottest spot is in southeastern China, with a weak second hot spot in the Middle East. In southeastern China, the frequency of the booze-inhibiting gene goes as high as 99%, with several populations in the 70% to 90% range. In the Middle East, the rates are more in the 30% to 40% range. Bing Su and his colleagues have brought these findings together with archaeological data on the origins of rice agriculture in East Asia, the transition from hunting and gathering to agriculture. The earlier rice agriculture started in a region, the higher the frequency of the booze-inhibiting variant of ADH1B in the populations now inhabiting those regions. Knowing the date at which rice agriculture began then allowed them to account for 50% of the variation in this gene's frequencies in Asian populations, which is amazingly high given the uncertainty in archaeological dates and all the other factors at work on these populations over thousands of years. Okay, fine. But what's the link from agriculture to alcohol? Well, broadly speaking, agriculture and the making of fermented beverages go together. Most hunter-gatherer populations do not have the means, know-how, or resources, for example, cereals, to make beer, wine, or spirits. Yet agricultural populations usually do, even small-scale semi-nomadic slash-and-burn agriculturalists. In China, the first alcoholic beverages date back almost to the very origins of rice agriculture along the Yellow River. About 9,000 years ago, in the ancient farming village of Jiahu, Chemical analyses indicate that someone had stored away 13 pottery jars of a fermented rice-based beverage that probably also contained honey and fruit. It seems that as soon as people domesticated rice, they quickly figured out how to make rice wine. Based on other historical episodes, this probably created alcohol-related problems for rice farmers, which favored any ADH variants that make drinking less fun. Without the cultural evolution of, first, rice agriculture, and second, rice wine, there might be no booze-inhibiting variant of ADH1B. Why some adult humans can drink milk. As with most mammals, drinking milk does little or nothing for the nutrition of 68% of the adults in the world. If you are a milk drinker, you are in the minority. Of course, whether they are humans or other mammals, all healthy babies come fully equipped with the enzyme lactase, which allows them to break down lactose milk sugars in their small intestines and thereby access the nutritional bounty in milk. Milk is a bonanza of calcium, vitamins, fat, proteins, carbs, and even water. In most people, the production of lactase wanes after nursing ends. By age 5, most people can no longer break down the lactose sugars in milk. Even worse, drinking milk often, though not always, causes diarrhea, cramping, gas, nausea, and even vomiting. This is lactose intolerance. In populations without access to medical care, such diarrhea can be deadly. However, in scattered populations around the world, including groups in Europe, Africa, and the Middle East, people can digest milk throughout adulthood. This lactase persistence allows older children, adolescents, and adults to access milk's nutrition. One study shows the global distribution of lactase persistence. Among indigenous populations from the British Isles and Scandinavia, over 90% of people are lactase persistent, while rates in Eastern and Southern Europe range from 62% to 86%. In India, the rate is 63% in the north, but 23% in the south. In Africa, the patterns are strikingly patchy. Some groups have high frequencies of lactase persistence, whereas neighboring groups do not. In the Sudan alone, rates range from around 20% up to 90%, depending on the ethnic group. In East Asia, lactase persistence is rare and often non-existent. Lactase persistence is under fairly direct genetic control and involves genes that inhibit the typical mammalian shutdown of the production of lactase after nursing ends. Though many factors have shaped the distribution of these regulatory genes, there are two key culturally evolved packages that have driven this bit of genetic evolution. First, it's only in the last 12 millennia that humans have domesticated animals like cows, sheep, camels, horses, and goats, which can potentially provide milk for adults to drink. So some populations adopted cultural practices that permitted them to keep animals and milk them. Such animals provide meat and hides, if nothing else. Initially, the extra milk would have been useful only for young children and infants. However, its presence would have created a genetic selection pressure for extending this lactose processing ability into middle childhood and beyond. Herding and milking are the first elements in the cultural package that selects for these genes. Crucially, these populations must also have persisted with herding and milking, but not have adopted the practices or culturally evolved the know-how for turning milk into cheese, yogurt, and kumis. Kumis is a fermented beverage made from mare's milk. While fresh whole milk from cows is 4.6% lactose by weight, cheddar cheese is 0.1% and tzatziki is 0.3%. Tzatziki is a traditional Middle Eastern yogurt-based dish. Some fancy cheeses like Gouda and Brie have only trace amounts of lactose. Thus, cheese and yogurt making are, at least in part, cultural adaptations for reducing lactose, which permit everyone to access much of the otherwise unavailable nutrition from milk. If populations had developed these bodies of technical know-how too soon, the selection pressure on genes for doing the same job would have been weakened. Thus, understanding who is, and is not, lactase persistent requires understanding both how cultural evolution can drive and how it can inhibit genetic evolution. Of course, many other factors influence when and where people can engage in herding, and thereby influence the strength of selection for lactase persistence genes. As with blue eyes, the region of northern Europe suitable for agriculture, but with limited UVB, may have created an especially strong selection pressure because of the calcium, proteins, and small amounts of vitamin D in milk. Calcium may inhibit the breakdown of vitamin D in the liver, and where it's cold, fresh milk can be stored for longer periods without having to turn it into cheese. Elsewhere, such as in the arid deserts of the Middle East and Africa, selection pressures for lactose persistence may have been increased by the water available in milk. Herders capable of drinking camel's milk, for example, might have had an advantage in traveling through arid regions or in surviving droughts. In some African regions, where herding might otherwise be rare or impossible due to extreme temperatures and diseases that plague herd animals, some societies developed cultural adaptations that involved systematically moving the animals to avoid temperature extremes and spacing their herds to suppress pathogen transmission. These populations, despite living in regions full of challenges to a herding lifestyle, are lactose persistent too, perhaps because of their locally specialized herding packages. Particularly interesting in this case is that natural selection found different ways to create lactase persistence in different populations. It appears that as herding animals became a centerpiece of economies around Eurasia and Africa, natural selection independently found 5 different genetic variants to suppress the shutdown of lactase production in different populations. In Europe, there was a flip on chromosome 2, just a short distance upstream from the gene that codes for the protein lactase, LCT. The DNA base cytosine got swapped for thymine. This flip may simply have thrown a monkey wrench into the otherwise mammalian standard off switch for the making of lactase proteins after weaning. Elsewhere, in Africa and the Middle East, the DNA swaps are different, though they are all between 13,000 and 15,000 bases upstream from LCT. Dating of the spread of these genes suggests that one of the African variants may be the oldest, with the European variants in the middle, dating to between 7,450 and 10,250 years ago. The variant centered in the Arabian Peninsula is probably more recent, between 2,000 and 5,000 years old. This timing suggests that the domestication of the Arabian camel may have created the selection pressures that favored this particular variant. The speed of this culture-driven genetic evolution is noteworthy. These selection pressures drove genes for lactose persistence to 32% of the global population in less than 10 millennia. That's really fast compared to the rates observed elsewhere in nature, and even in the human genome. Before moving on, it's worth highlighting the downside to not recognizing that such culture-gene coevolutionary processes have occurred and are occurring. The ill effects of promoting milk drinking among peoples without lactase persistence didn't begin to dawn on American researchers until 1965. Prior to this, Americans assumed that if drinking cow's milk was good for our European descent children, it must be good for everyone's children. In 1946, the National School Lunch Program required that fluid milk be part of any student lunch funded by the program. Despite the growing scientific literature, government efforts continued to promote milk drinking for everyone into the 1990s. As late as 1998, the then Secretary of Health and Human Services even appeared in the well-known Got Milk commercials. Over the years, many of the famous sports and music stars who appeared in these commercials donning milk mustaches were not, in all likelihood, actually able to digest the very beverage they were promoting. Culture gene revolutions. These cases of culture-driven genetic evolution are three of the best documented examples we have, though there's every reason to suspect that they represent only the tip of an iceberg. The evolutionary biologist Kevin Leyland and his collaborators have already fingered over 100 genes that have likely been under selection, based on analyses of the genome, and have at least plausible cultural origins. These genes influence an immense spectrum of traits, ranging from dry earwax and malaria resistance to skeletal development and the digestion of plant toxins. What these cases illustrate for our purpose is as follows. 1. Culture can exert a powerful force on genes, driving genetic evolution. Gene culture packages emerge and spread rapidly, as with milk drinking, blue eyes, and booze avoidance. 2. In fact, the selection pressures created by culture can be among the most powerful observed in nature, and broad genetic sweeps can occur in tens of thousands of years. Culture gene coevolution can be remarkably fast. 3. We can point to specific genes on particular chromosomes, and sometimes even know which molecular base changed. Once hypothetical genes have now been pinpointed. 4. Once cultural evolution creates the selection pressures, natural selection often manages to find and favor several different genetic variants to address the challenge. 5. However, sometimes cultural evolution can sap the strength of selection, as highlighted by the populations who rapidly developed cheese and yogurt-making technology. One concern with the aforementioned examples is that all stem from the emergence of food production, from agriculture and animal domestication. Perhaps this major revolution in human history is a unique event from which we cannot draw general conclusions. To the contrary, my view is that the agricultural revolution just happens to be the best timed revolution in order for us to detect its causes and effects in our genome. The Industrial Revolution is too recent, and the revolutions that preceded food production are much older and thus harder to study. Nevertheless, there's every reason to suspect that there was a cooking and fire revolution, a projectile weapons revolution, and a spoken language revolution, among many others. And as you will learn in later chapters, technologically driven revolutions are probably underpinned by revolutions in forms of social organization or institutions. The agricultural revolution is just the one in the temporal sweet spot for today's science. To see something of this, consider that chimpanzees have 2 copies of the AMY1 gene, but humans have, on average, 6 copies. The gene codes for a protein, amylase, found in saliva that helps break down starch. The extra copies mean that humans, on average, end up having 6 to 8 times more amylase in their saliva than chimpanzees. All other things equal, this means we are better at starch processing than chimps. So, after you beat that chimp in a marathon, you should challenge him to a potato digesting contest. Human populations, however, vary in the number of AMY1 copies they have. Populations who have long been dependent on eating high-starch diets have between 6.5 and 7 copies, on average. The Hadza, African hunter-gatherers who live in savanna woodlands and rely on starchy roots and tubers, have the most, at almost 7 copies, on average, and some Hadza have as many as 15 copies. European Americans and Japanese are not far behind, at 6.8 and 6.6 copies. By contrast, populations long dependent on low-starch diets have copy counts around 5.5. These include other African hunter-gatherers who live in the tropical forest of the Congo Basin, and herders in both Africa and Central Asia, who depend primarily on some combination of meat, blood, fish, fruit, insects, seeds, and honey. These differences are likely part of a long and meandering evolutionary story, and emerged as our ancestors shifted to a heavy reliance on underground roots and tubers over 1 million years ago. However, just how much populations have depended on starch since then has been influenced by a combination of ecology and cultural evolution, including the practices, preferences, technologies, and know-how of different populations. As we see from the examples, groups can live relatively close by in similar ecologies, but still maintain different numbers of AMY1 genes because they operate with different economic packages. There is also evidence that culturally prescribed forms of social organization can shape our genome. This is important, since some have argued that the forms of social organization created by cultural evolution are too weak or unstable to affect our genes. One important aspect of human social organization is what anthropologists call postmarital residence. In many human societies, especially until recently, local norms specified that a newly married couple went to live either with the husband's family or with the wife's family. The first is called patrilocal residence, and the second, matrilocal residence. Working in 3 patrilocal and 3 matrilocal farming populations in northern Thailand, Hiroki Ota and his colleagues examined the variation in people's mitochondrial DNA and Y chromosomes. Both sons and daughters get their mitochondrial DNA from mom, and only mom. Sons get their Y chromosomes from dad, whereas daughters don't get Y chromosomes at all. If social organization is stable enough to influence the genome, then patrilocal communities should have relatively low variation in their Y chromosomes compared to mitochondrial DNA, because sons always stick with their fathers. Similarly, because daughters stick with their mothers, matrilocal communities should show the opposite pattern: low variation in mitochondrial DNA and higher variation in Y chromosomes. This is precisely what Ota's team found, showing that culturally evolved social norms can shape the genome. Overall, cultural evolution can, and has, powerfully shaped the human genome in a variety of important ways. As we saw in Chapter 5, this culture-gene coevolutionary interaction goes well back into our species' history, where culturally transmitted know-how about fire, water containers, tracking, and projectiles were some of the key selection pressures favoring aspects of our anatomy and physiology. Moving forward, I'll begin to focus on how culture created selection pressures on genes that influence our psychology and sociality. In Chapter 7, we'll take another step by going deeper into the subtle and nuanced ways cultural evolution can build adaptations without the culture bearers themselves having any idea what's going on. Genes and races. Before moving on, it's worth highlighting a point about genes and race. Anthropologists have long argued that race is not a biological concept. What we mean by this is that the racial categories developed historically by Europeans, such as Caucasian, Negroid, and Mongoloid, do not convey or contain much, if any, useful genetic information, aside from capturing something of the migration patterns of ancient peoples. Detailed studies of the genome, including the research mentioned, has only served to further underline this point. As we discussed, skin color genes are heavily influenced by a combination of UV radiation and diet because they affect vitamin D and folate. This means that people in New Guinea and Africa are both very dark-skinned despite being from opposite ends of our species family tree. And very light-skinned Europeans are evolutionarily recent, being mostly a product of agriculture at high latitudes. Other genes have quite different distributions for distinct reasons. For example, we saw that lactase-persistent genes are common among indigenous populations in Britain, and some African groups exist at moderate frequencies among Eastern Europeans and Middle Easterners, and remain at low frequencies in other African groups and many Asian populations. Similarly, amylase genes are more common among Japanese, European Americans, and Tanzanian foragers, but less common among Congo foragers and herders in both Tanzania and Central Asia. What does race tell us about these genetic differences? Nothing. Traditional racial categories just don't tell us anything about this important variation. In fact, the processes I've described actually make classical racial categories even less informative, since they operate in diverse and non-concordant ways within races to make local groups less similar. For example, lactase-persistent and non-persistent Africans, while at the same time making different continental races more similar. For example, amylase genes in Japanese and Americans. The current evidence indicates that natural selection operates in diverse ways on scales much smaller than races and simultaneously on different continents. Moreover, even using categories, racial or otherwise, often distorts the picture. The genetic distributions on these maps vary continuously, so it's best to forget about discrete boundaries. Overall, traditional racial categories capture only about 7% of the total genetic variation in our species, which reveals that races are nothing like the subspecies found in chimpanzees. Given our global distribution and range of environments, our species' genetic variation is actually rather limited. Of course, this is not surprising when you realize that in addition to sometimes driving genetic evolution, cultural evolution can also inhibit genetic responses by more rapidly generating cultural adaptations of the kind discussed in Chapter 7. For good historical reasons, many people are sensitive to scientific and evolutionary research on genetic variation, especially variation among populations. In the last century, pseudo-scientific efforts to formalize folk concepts of race were used to justify much violence, oppression, and even genocide. However, two rapidly developing areas of research ought to, somewhat, allay concerns about the return of pseudo-scientific racism. First, our understanding of human genetic variation, derived from studying actual genes, completely dismantles any remaining shreds of the old racial notions, as the examples show. The best antidote for pseudoscience is real science. Second, psychologically oriented researchers have come to increasingly understand how and why humans are susceptible to lumping people together in labeled groups and slapping stereotypes on those groups. As you'll discover in Chapter 11, racial and ethnic categories arise when cultural evolution taps our human universal tribal psychology to carve up the social world in particular ways. Though these categories are not usually rooted in any important genetic variation, these categories are learned unconsciously and can affect our perceptions, automatic intuitions, and rapid judgments. Increasingly, we understand what triggers prejudice and what the implications of this is for health, education, economics, conflict, and social life. What we need is more evolution-grounded science on genes, culture, ethnicity, and race, not less. These insights will continue to fuel the spread of a new social construct, the view that all people, and perhaps some other species as well, are endowed with certain inalienable rights. We call these human rights. No new facts about genes, biology, or culture can alienate a person from these rights.